And I'd like to start by introducing Nick Croucher, who's come here from the Sanger Institute in Cambridge. The other Cambridge.
Thank you, Bill. Yes, as you said, I'm Nick Croucher. I was a PhD student at the Sanger Institute up until 5 days ago, and I'm here to talk about some of the inferences that we've made about the evolution of drug resistance in Streptococcus pneumoniae following some of our genome sequencing projects. So to briefly introduce the pneumococcus, it's a gram-positive obligate commensal of the mammalian nasopharynx. Its genome is about 2 megabases in size, and has a firmicute, has a GC content of around 40%. And the most interesting strains clinically are those that are encapsulated. So there are more than 90 antigenically distinct capsule types or serotypes, and this is important for immune evasion that allows it to cause systemic disease such as pneumonia, sepsis, and meningitis. The capsule is also the target of many vaccines over the years. Most recently, the polysaccharide conjugate vaccines, of which the original formulation, PCV7, the heptavalent one, targeted 7 of those 93 or so serotypes. Bacterium is also naturally competent, so it's able to take up exogenous DNA from the environment and integrate it into its own genome at regions of sequence similarity. So I'm just going to talk to you today about 2 different lineages with quite contrasting patterns of evolution. And the first of those is the pneumococcal molecular epidemiology network clone 1. So this was one of the first multi-drug resistant pneumococcal lineages to become a global problem. It was first detected in Western Europe, in particular UK and Spain, in the early 1980s, and then by the early 1990s was found to have transmitted to North America, where by the end of that decade, it was causing about 40% of penicillin-resistant pneumococcal disease. Around the same time, it's found to be causing about 30% of pediatric penicillin-resistant pneumococcal disease in South America, and roughly contemporaneously, it was found to have disseminated to South Africa and Southeast Asia as well. Most strains have a serotype of 23F, which is one of the 7 serotypes included in the PCV7 vaccine. And so levels of disease caused by that have dropped off as the vaccine has taken effect. However, this fall has been somewhat offset by an increase in disease caused by the non-vaccine serotype 19A. And a small proportion of that has been due to serotype 19A variants of this PMN1 lineage. So our first step at the Sanger, as ever, is to sequence a genome. The first one we did was the type strain isolated from a hospital in Barcelona in 1984, a 2.2 megabase genome. The reasons behind the penicillin resistance have long been known in the pneumococcus, resulting from modified penicillin binding protein sequences that have a lowered affinity for beta-lactams. But in this particular lineage, it wasn't known precisely why it was resistant to chloramphenicol and tetracycline as well. And that happened to be due to this large integrative and conjugative element you see in this 7 o'clock position in the genome. So just to look at the details of that, it's an 81 kilobase long element. It's actually a hybrid consisting of this small TN916 element inserted into the larger TN5252 backbone. And it's the TN916 that contains the TETM tetracycline resistance gene, whereas the TN5252 contains this linearized integrated plasmid PC194 that's originally found in Staphylococcus aureus. So that gave us an idea of the structure of the genome and the characteristics that were shared by all members of the lineage. To look at how it's subsequently expanded its repertoire of antibiotic resistances and how it's gone on to respond to the vaccine selection pressure, we used a much larger collection of isolates. We, in total, gathered 240 strains from our kind collaborators in Europe, America, Asia, and South Africa, from 22 countries in total, spanning 24 years. So we sequenced all of these as multiplex libraries on the Illumina platform, which gave us a large amount of raw short-read data for each of them, which we then aligned to our complete reference genome, which you can see sort of down the bottom here. And this is what the raw reads look like aligned to that. The bases are blue when they are in agreement with the reference genome, and they're red when either they're sequencing errors over here, or as you can see, this long, this tall column of mismatches when there's SNPs. So using those SNPs, we can construct a maximum likelihood phylogeny of all the strains in our collection. However, this initial phylogeny is simply not very good. And the reason behind that is very clear when you look at the pattern of polymorphisms distributed around the genome. What you get are single lone SNPs, and then some in very dense tracts. And we can interpret that as point mutation and then recombination, importing diversity from outside the PMN1 lineage. So what we wanted to do to generate a better phylogeny was to develop a method that could use just these point mutations, just the clonally descended parts of the genome, to construct the relationships between strains and avoid any distortion by the bits of the genome that were affected by horizontal sequence transfer. So we did that in an iterative algorithm that detected SNP-dense regions and generated this phylogeny, which we are much more confident in, because when you look at the correspondence between the distance from root to tip and the date of isolation of strains, it predicts that the lineage originated about 1970, and we reconstruct that it originated most likely, given our sampling, in Western Europe, and this fits from the epidemiological observations that I described earlier. We can start to tell some of the phylogeography from this pattern, from this tree, so we can see a very successful intercontinental transmission here to South Africa, where there appears to have been a single bacterium that passed across and has given rise to a large population of drug-resistant bacteria. On a smaller scale, we see a similar thing in Vietnam, and that most of the isolates from Southeast Asia are mixed into this large, widely disseminated clone, where we also see representatives from North America and Turkey and Russia. We can also get some information about the recombinations that have happened that are picked up by our algorithm. So the same tree here on the left. We have the reference annotation I showed earlier in simplified form across the top. And then in this dotted panel here, we have a row for each taxon in the tree, which shows a colored block where we predict recombinations to have occurred. So they're colored red when they're reconstructed as having occurred on an internal branch in the tree. So they're shared by multiple isolates by common descent, or they're colored blue when they're predicted as occurring on a terminal branch of the tree, so they're unique to only one of the taxa. And having looked at this, we can then look at the density of recombination events across the genome, and we find that it's these important antigens, such as pneumococcal surface protein A, that are hotspots for detected recombinations, and of course this CPS locus, which is actually the locus that determines the capsule type and therefore serotype of the bacterium. And these recombinations here match up with the changes from the ancestral 23F serotype. So we get individual changes to serotypes such as 14 and 3, but with 19F and 19A, we see 3 independent changes to each of those serotypes during our sampling period. This bottom one to 19A is the most interesting one because these represent strains from the USA that emerged post-PCV7. So just to look at all of the American strains, this widely disseminated clone accounted for the majority of them in the era before the vaccine. They're both 23F and 19F serotypes, which are both included in the PCV7 vaccine. And then following its introduction in 2000, gradually after 2005, we get these green and serotype 19A non-vaccine serotype strains emerging. And what you can see is that you've gone a population shift from these strains here all the way down to these, which we didn't detect prior to the vaccine. So it appears to be a sufficient selection pressure that results in a previously undetectable subpopulation rising to prominence. So this implies that there's quite a rare event to escape a vaccine, at least in this lineage. So how did this compare to drug resistance? The simplest drug resistances to look at are those that involve a single SNP, a single base change in the genome. So for rifampicin, for instance, we actually see 5 different independent changes throughout the genome that lead to resistance, most of them in South Africa, where you'd expect rifampicin to be used quite frequently in treatment of tuberculosis. More strikingly, you can see a similar pattern for fluoroquinolones, where we have 26 independent base changes associated with fluoroquinolone resistance occurring throughout the tree. These may be somewhat unrepresentative, though. They don't look like the vaccine escape. They look like they're very frequent, easy mutations, but they are only point changes. Perhaps more representative is the problem of macrolide resistance, which is more commonly caused by the acquisition of cassettes rather than the occurrence of single SNPs. So through de novo assembly of our data, we found that there were 3 different types of macrolide resistance cassettes in the PMN1 lineage. 2 of them, the omega element and the TN917 element, both carry these ERM-B genes, which are methylases that modify the ribosomal RNA that's the target of this antibiotic. The mega element instead has a mel-meth ABC efflux pump that removes the antibiotic from the cell. So we have multiple different cassettes, and we find that they actually all integrate into a similar place, but they're found in multiple different arrangements. So this is the TN916 element I showed you earlier, which has the TETM resistance gene. And this is from our reference strain back in 1984, which is macrolide sensitive. And we find that all of these TN916 elements from different isolates in our collection, when you compare them with these red bands representing blast matches, have many different insertions in them, which correspond to the 3 different macrolide cassettes. And you can see that they actually occur all in different arrangements, with many TN916 elements having more than one macrolide resistance cassette. So we have multiple cassettes, multiple arrangements. We find they all come into the come into the lineage multiple times. So this shows a mapping coverage heat map for all of the taxa in the tree. So each one has a row, and when you map the raw Illumina data, red indicates a high level of coverage, and blue indicates a low level of coverage. All of them have this TN916 element, and then we have the 3 different macrolide resistance cassettes that you see seem to enter the population quite frequently. Quite interesting is this block here at the end, where a group of strains appear to share the mega cassette through common descent. And what we see is that that mel-meth system is supplemented with an ERM-B gene, and in one case, a TN917 cassette is actually acquired, and the original mega cassette deleted. And this might correspond with the extended spectrum of resistance that results from the ERM-B mechanism of resistance rather than the efflux mechanism. So over a few decades, we have a single lineage that essentially appears to be able to pick and choose which macrolide resistance cassettes it wants to have. The mechanism involved is usually direct integration of the macrolide resistance cassette into the TN916 element. However, sometimes it's a wholesale replacement of the entire integrative and conjugative element. So here we see one of the strains where, again, the blast matches are indicated as red bands unless they're in the inverse orientation, in which case they're indicated by blue bands. And you see this whole 81 kilobase island is replaced by a different one in this strain, and it retains the same basic structure of having a macrolide resistance cassette inside this TN916, inside a TN5252. And again, a separate strain, you see the same kind of thing. So it's not just macrolide resistance cassettes that appear to be quite readily available to this lineage. It's also entire integrative and conjugative elements that carry chloramphenicol and tetracycline resistance as well. However, it's not fair to characterize the evolution of PMN1 as an inexorable increase and expansion in its antibiotic resistance. We also see that it's lost. There's an equilibrium process of both acquisition and deletion. Here, for instance, in the omega element is most frequently seen in this form, which results from the deletion of this aminoglycoside phosphotransferase that sits between 2 different omega repressor tandem duplication genes, and when these excise, they leave a small scar behind. So this isn't that surprising, given that kanamycin is something to which pneumococcus has an intrinsic high level of tolerance. Another antibiotic to which it loses its resistance is actually chloramphenicol. So this is something that has undergone a decrease in use over the years in the West, and we see 2 Western European strains where this here represents a de novo assembly of the strain's integrative and conjugative element. You can see that it actually has deleted this large chunk here, and this corresponds to the chloramphenicol resistance gene. So just to confirm that, we mapped back the raw data onto the referenced chromosome, and you can see again that all the data from this section here is missing. So we have a second example of that, again from some West European strains, where we have this large deletion, including the chloramphenicol resistance gene. And over here in the TN916 element, we actually see that the coverage of the genome doubles, which suggests that it's acquired a second TN916 element somewhere else in the chromosome, which again emphasizes just how readily available these elements appear to be. So that is enough about the integrative and conjugative element. What about the penicillin binding proteins that determine the beta-lactam sensitivity? Again, we see a similar picture to that which we saw for the other antibiotic resistances. There's 6 PBPs in the genes in the pneumococcal genome. 3 of them are clinically relevant for resistance. we see that this PBP1A, which is important for both penicillin and cephalosporin resistance, is quite tightly conserved. It only has one recombination that affects a small part of the gene. But PBP2X has undergone actually a total of 12 recombinations affecting some part of the gene in the data set. And different alleles of this gene are known to be associated with different levels of cephalosporin resistance. So this may, again, correspond to a resistance determinant being very readily available to this genotype. It can actually acquire, as it diversifies and spreads throughout the world, acquire many different versions of this resistance allele. So to summarize, what we're looking at is a PMN1 strain that is diversifying primarily through recombination. It's accounting for 88% of those SNPs. But we're looking at something where the SNPs causing resistance are frequent, but so are the acquisition of macrolide resistance cassettes, so is the acquisition of conjunctive elements, and so is the acquisition of PBP alleles that cause resistance. It seems to be able to just soak up a very wide and diverse range of genes from the environment that cause resistance, although we do also see some loss. So we have got this rapid flux, whereby the strain is reaching equilibrium according to its selection pressures. So for something completely different, serotype 3 is another pneumococcal, well, sequence type 180, serotype 3 is another pneumococcal lineage. It has a very thick mucoid capsule. You can see on the left here, these are unencapsulated isolates, and then these are the versions with the capsule, just grown on an agar plate. It has a very clonal population structure. So again, as with this PMN1, we sequenced a complete genome as a reference, this time from a case of asymptomatic carriage in Oxford. And we took a collection, this time of just around 80 strains, primarily isolated from Europe and USA, and performed the same kind of analysis as we did for PMN1. And what we see here is that it's a very different story. Rather than there being a lot of diversity all the way through the tree, we have a single long branch separating these two strains from the rest of the population, and that is where almost all the diversity in the lineage is found to be. Within this clone clade here, we find that all strains have a very similar pattern of recombination. It's a very stable genotype. We also see in this clade no evidence of any conjunctive elements coming in. And perhaps most surprisingly, not even the point mutations, the SNPs that led to resistance to rifampicin or fluoroquinolones, don't necessarily involve any horizontal sequence transfer. These also don't appear in this clade, with a single exception, which corresponds to these two strains down here. What's interesting about these two strains is they both come from the same case of adult meningitis. One of them was isolated from the blood and one from the cerebrospinal fluid. And they're distinguished by just 3 polymorphisms, all of which are of the ancestral allele in the blood strain and the derived form in the cerebrospinal fluid strain. One of those polymorphisms exists upstream of this PAT-A, PAT-B, a putative antibiotic transporter operon. What we see is when we characterize these two strains functionally using RNA sequencing to quantify the level of expression, is that this dark red line here indicates transcription of this bottom strand where the functional genes lie in the blood strain. And then on the dark blue line here shows the level of expression in the CSF strain for the same genes. And you can see that before, upstream of the SNP, there's very little difference in the levels of expression, but downstream there's a 4-fold upregulation of this pump that leads to the acquisition of a ciprofloxacin and the fluoroquinolone resistance phenotype. And this appears to have happened within the patient. So the polymorphism, you can kind of ascribe a function to that because it weakens a hairpin in the leader sequence, so it seems to be a relatively simple regulatory mechanism. So serotype 3, in very much contrast to PMN1, has a very stable genotype. There are very few recent recombinations that would introduce penicillin resistance. There's no acquisition of conjunctive elements that would indicate resistance to other antibiotics. And there's relatively few SNPs that seem to lead to rifampicin or fluoroquinolone resistance, except in one case where selection appears to have had the least time to act. So just to summarize and finish, there are few barriers to the development of drug resistance in lineages such as PMN1, it would seem. As I said, recombination, conjugation, point mutation, direct integration. There are multiple instances of all of those in the sample that we had. The persistence of this resistance There's the drug, kanamycin, and chloramphenicol, where the selection pressure is weaker. They seem to be lost relatively frequently. Other ones, such as macrolides, you get acquisition of one cassette, two cassettes. As I said, it seems to be able to pick and choose which one it wants. So within the pneumococcus, we are looking at a wide range of evolutionary strategies. Here are presented two extremes. We don't know whether everything will fit as a spectrum between those two, or whether they will fall either into one camp or the other. But both appear to be able to be successful, whether it's multi-drug resistance or whether it's almost universal susceptibility to all antibiotics. And I look forward to hearing about why that is in the next 3 talks in this session. So it just remains to me to thank everyone who's been so helpful with this project, both at Sanger and externally, and providing strains, to thank the Wellcome Trust for funding me, and to thank you for listening.